Hi, this is Mrs. Murdock, and I'm going to talk about action potentials and resting potentials today as they occur on a neuron. So I've drawn a picture of a neuron here, and the first thing I want to remind you is that this really is a cell transport problem. So things are moving across membranes here. So the inside of this cell is the cytosol, and I made the axon. This whole thing is the axon here. I made it big and fat so you can see what's really going on in there. And you've got the dendrites over here that are receiving signals, and then you have electrochemical signals that travel along the axon and then are delivered to the synapse at the end. And that signal might go to another neuron, or it might go to a muscle cell to tell it to move, or it might go to a gland to tell it to secrete something, right? But we're going to concentrate on what's happening along the axon. So first of all, let's talk about resting potential and what resting potential is. Now again, I don't have this diagram printed out for you, so you don't, if you want to draw it, be my guest. But I'm also recording this so that you can see it later. So resting potential is actually kind of a misnomer, because resting potential takes energy to maintain. So with resting potential, the outside of the membrane has kind of an overall positive charge, and the inside, the cytosol, has an overall negative charge. And this is true along this membrane and also down here. You also have, here's the inside of the cell is slightly negative and the outside of the cell is slightly positive. And that is a gradient. That's a difference, and we learned that gradient meant difference. And really, anytime there's a gradient in nature, you should remember that nature doesn't like that. And it actually takes work to keep things different, because nature wants everything to flow back so that equilibrium is there again, right? So what the neuron is doing here is it's kind of like it's pulling back on a spring, which takes energy, right? So that it's ready to receive a signal, receive a stimulus, and send it, okay? So the way that it maintains this is it uses a particular type of protein, membrane protein. Remember, this is a membrane, phospholipid bilayer, just like any cell membrane. And embedded in that membrane are proteins. And some of them are these, didn't change color for me, excuse me. Some of them are these red ones here, okay? And the red ones are sodium-potassium pumps. I'm going to draw them in red down here. Sodium-potassium pumps do exactly what they sound like they do. They pump sodium and potassium. They send 3 sodium ions, which you remember are positive, out of the cell, right? Out of the cell. And at the same time, 2 potassium ions, they bring them in to the cell. Okay. So what's happening here? So you have the 2 players, the sodium ions and the potassium ions, are moving in such a way that you have sodium ions out here in pretty high concentrations at resting potential, and inside you have potassium ions in here. Okay? And a lot of students get confused about this, because then where is the negatives coming from if all the ions that are moving are positive? So what you have to remember is that that's not the only ions that are in the cytosol of cells. You also have chlorine ions in here, which are negative, and you have proteins that very often have negatively charged surfaces because, remember, the polar R groups are facing the water. Okay, so you have lots of other ions and things dissolved in here that are negatively charged. So if you push a bunch of positive ions out, more out than you push in, the relative charge is going to be negative in the cell. Okay, and if you happen to have a really tiny little voltmeter, which is, I'm going to show you this over here. Here's a voltage meter, and there's some sort of marine animal that has such huge axons that you can put a little sliver of a voltmeter in there and actually measure what the charge is inside the cell at different times. So at resting potential, the measurement, I think you can see it if I pull this up, the measurement inside the cell, because of all those chlorine ions and other negative things, and because we pushed a bunch of positive stuff out, resting potential is negative 70 millivolts. So that's our first active measurement of what's going on in the cytosol, inside the cell, okay? So we'll keep track of that. So resting potential, a gradient, maintaining that gradient takes energy, and it's the sodium potassium pumps that do that, and that is an active transport process. It's one of the very few that we've talked about, really. We haven't talked a whole lot about active transport. Here's a really good example. The sodium potassium pump preparing a neuron cell for a stimulus. Okay, so now the second step is, and this is where it gets difficult for me to draw stuff, but let's say that there is a stimulus over here that hits this side of the cell where the dendrites are. So the stimulus hits it hard enough to cause these green proteins, which is a different type of protein. These green things, okay, those are sodium gated channels. Gated channels. They are not pumps. They are proteins that are just for sodium, and they open to let sodium flow into the cell. Into the cell. I know you guys are writing furiously, but if you can just answer me for a moment, would that be a passive or active process? What do you think? Passive, excellent, yes. So this is really weird, isn't it? That the resting potential is an active process and the action potential is actually passive. The door opens and the sodiums flow down along their gradient to reverse the polarity of the inside of the cell. That's what an action potential is. So action potential is actually passive. It's passive transport of all the sodiums that are on the other side, outside the membrane, waiting to get in, and this is their big chance, right? So the sodium is going to flow in and completely reverse the polarity along a slug that kind of moves along like this. That electrochemical signal, electrochemical signal is going to move all the way down to the end of the axon and the terminal over here, the terminal, where it can then jump and signal something else. But that chemical signal is just a reversal of charge. That's all it is. So that now we have positive charges inside the cell because all that sodium flowed in, and now it's negative outside. And this is action potential. This is action potential. And this is why I had that video earlier, because I find this difficult to draw. So that slug of action potential moves all the way down, and it actually goes really fast. I mean, think about this. When you put your hand on a hot stove, are you waiting for Mrs. Murdock to draw a slug of action potential down? No. It's like, like that. It's really fast, right? So a nerve signal travels faster than you can even say action potential. And it's made of salt, right? Basically, sodium flowing in. Okay, so without making that any more complicated, let's go down here. All right, so that action potential, if you now measure the inside of the cell, right, when that action potential is going by, the action potential millivolt reading, what do you think it's going to be at its peak? It's going to be a positive number, and it goes to about plus 30 millivolts at its peak. And that's due to sodium flow into the cell. all that positive charge coming into the cytosol raises the charge from negative 70 all the way up to positive 30 millivolts, okay? So that's the peak of the action potential. Now the problem is, you can't leave the cell like that because it has to get ready again to receive another signal. You have to reset it. Whatever you do in biology, you have to undo it to get ready for the next thing, right? Okay, so now behind the action potential, the potassium, which I think I made blue, the potassium also has its own gated channels. So the potassium gated channels now are going to open. They don't open until the sodium has finished getting up to positive 30. And they open to remember the potassium was inside the cell, so it's going to flow out to let potassium ions flow out of cell. And again, this is diffusion. It's going down its gradient, right? Down its gradient to restore or repolarize, right? Repolarize the membrane. Okay. Even go over here. All right, so that's the sodium flow. Now, we're going to go repolarizing. Repolarizing with the K plus ions, actually, it overshoots it. It doesn't just go to negative 70, it actually goes all the way to negative 80 millivolts. So it takes it a little bit further past the threshold. And then you have a problem. You have a problem here because, yes, you've stopped the action potential behind it. You're resetting it back to where it was, but the ions are all in the wrong places. So the sodiums are now all inside and the potassiums are outside. We got to switch them back. So what do? Well, first of all, let me explain the problem. Okay. The Na and K plus are in wrong places, right? They're not in the right places. They're in the wrong places. So what has to happen? Yes, the sodium-potassium pump's beautiful. The sodium-potassium pumps back to active transport, right? Reset the resting potential and put all of the potassiums and sodiums back where they need to go. Okay, and once again, they have to use energy to do that. Right. Okay, now before I run out of time here, I want to put this on a graph for you because it's the graph that I'm really going to be asking you about next Thursday, okay? So if I were to, that little voltmeter, remember, that was measuring what's going on in the cytosol, the resting potential is down at negative 70 in the beginning, right? And then there's a stimulus, and what happens? Why is it going up? Which thing is flowing? Right, the sodium is flowing. Very good. So the sodium ions... I heard somebody say it. Okay, sodium ions are flowing in. It is depolarizing, right? Hits 30, and the potassium gates open, and you go back down because potassium is flowing out, and it's repolarizing, and then it hyperpolarizes for a little bit, and then goes back to resting. There. Okay, so let me put those words up here because those are useful. When this is going up, what's really happening here is depolarization. Depolarization on the way up. Sorry for my spelling. Depolarizing because it was polarized and you're unpolarizing it. And then back up here, you're repolarizing it. Right? Going back down. And this is hyperpolarizing because it goes a little too far. And then back to resting potential. Okay? All right, that is the story of the action potential, and I hope that that is helpful.